There. All right, so now we should be at the beginning of this. And I was telling people just a minute ago, Randy and I have had a discussion about physiology of diving and oxygen consumption, and energetics and hypoxia and transport of oxygen going on 40 years now. And sometimes we agree, sometimes we don't agree, but we thought it would be fun to go over some of the concepts that we've been discussing and bring it into the era of COVID also, which was the nature of a paper that we did. So we're going to be looking at aerobic metabolism when gas exchange stops in these marine mammals, but also sort of venture into disease. And the information from this talk you can find in a paper that Randy and I co-authored regarding physiological resiliency in diving mammals. And then also in his book that has come out just in the last year or so on marine mammals and adaptations for aquatic life. And we're more than happy to talk to any of you. You're all colleagues in one way or another. And in terms of how we sort of started this, we went back to the Taylor and Weddell sort of pathway for oxygen. And it gave us sort of a mechanism that allowed us to hang all this information about energetics and use of oxygen in the body of an animal by just compartmentalizing things. So we knew that we had air coming into our mammal. It was going to be going through the respiratory system. That was going to have to connect to the cardiovascular system, all the blood vessels, all the changes in heart rate that were going to occur between rest and activity. And then eventually we were going to have to get that oxygen to the cells and the various organs of the body. And this obviously is a terrestrial mammal, but the truth is the major players in what we're talking about are the same for marine mammals with a little bit of a variation. And this was in the paper that I had showed you previously. And it shows the same kind of pathway for oxygen within our breathing animal when it's on the water surface. This is a beluga whale, obviously, but it's the same idea. You've got to have air coming in. It's going to go through the respiratory system. That's got to be matched in terms of the movement of gases to the cardiovascular system, and that's going to get to the cells of the around the body. The difference, though, is that here's what's happening with the animal loading oxygen when it's on the water surface, and it's going to go on a dive, as marine mammals do, and things are going to change within that pathway in terms of lung collapse that can occur, changes in the vascular system, the whole dive response in which you've got a bradycardia set up, and changes in peripheral vascularization. And then this sort of black box that's here in terms of what's happening as far as organ metabolism and organ function. And I've left it open in this case because a lot of times we're talking about locomotor energetics, and that's still happening. You know, an animal when it's taking a stroke is going to use ATP. There's going to be an oxygen need there. And so you've got a marine mammal when it's exercising during the course of a dive, going into a situation like this with the dive response, and then coming back up to the surface to do all of this oxygen loading. And the thing is that they're doing this over and over and over again. This isn't a one-time event. You know, we always teach it as a one-time event. But in actual fact, these animals are exercising at some deep dives or shallow dives or a series of short dives when animals are transiting. But these events along the oxygen pathway are happening sequentially, and they're happening over and over and over again during the course of the day. So with that background, I'm going to let Randy take over in a second here and start to tell you about the energetics. How much oxygen are we really talking about in terms of these animals moving it through the body, and how does it change as the animals are diving? So I'm going to stop my share, bring you all back, and now Randy, you can go on for the next part of the talk.
Okay, so the pathway for oxygen, that Terrie just laid out is there for a reason, and it's to provide oxygen to the cells through a respiratory system and a cardiovascular system. And this has evolved over time to support a level of metabolism that includes resting metabolism, and it also includes metabolism associated with activity. If we look just at resting metabolism, endothermic animals, such as birds and mammals, have a resting metabolic rate that roughly scales allometrically. And here's some data from McNabb, a wonderful data set, showing these dark spots are terrestrial mammals. And if you scale this, you can see that the scaling factor is less than 1. So it's not isometric, it's allometric. And it falls very close to what we call the old Kleiber value of 0.75. Actually, it does vary a little bit depending on which species of animals you're talking about. I will point out, though, that around this line, there's a fair amount of variability, which is probably adaptive within species. That is, this scaling coefficient to body mass is not set in concrete. It's not the same for all mammals. There is some variability around this mean scaling factor. If we look at carnivorous marine mammals, what we find is consistently a resting metabolic rate that is greater than the allometric prediction. As a group, they consistently are higher than what we would predict in a terrestrial mammal of the same body mass. With the exception of the manatee, which is herbivorous, and I'm not going to talk about the manatee today, but it has one of the lowest resting metabolic rates that we've ever recorded. And that's an interesting story in itself. But I'm going to focus on the carnivorous marine mammals, which includes sea otters, Pinnipedia, and Cetacea. This level of resting metabolic rate is 4 to 8 times higher than an ectotherm, a reptile. And that additional heat production is what allows endothermic birds and mammals to maintain a high and constant core body temperature. And that extra heat, we believe, comes from a mitochondrial proton leak, which I'll talk about in a minute, in which additional heat is produced in the mitochondria by basically releasing the electromotive force across the inner mitochondrial membrane as heat rather than generating it as ATP. So here again is a plot showing some marine mammals here on this dashed line, and our Kleiber curve, which is very similar to the data for McNabb that I showed you earlier, showing this consistent elevation of resting metabolic rate, which ranges from about 1.4 to 2 times higher than the allometric prediction for a large group of terrestrial mammals. So these animals have an elevated metabolic rate. And we think it's a thermoregulatory adaptation that's necessary for these animals to offset the increased thermal conductance of water, which is 23 times greater than air. So this is an adaptation for living in water with an environment with a very high thermal conductivity. And we see it in almost all marine mammals, except for the manatee, which is a herbivore and actually is restricted to very warm water and can't actually even tolerate cold water or cool water. If we look at this as a group, here are a number of marine mammals of varying body masses in grams. And the measured basal metabolic rate or resting metabolic rate, the predicted from our terrestrial mammal regression, what we see is consistently an elevation over the allometric prediction, with the highest being the sea otter, almost 3 times higher than the allometric prediction, one of the highest resting metabolic rates for any mammal that's ever been measured. If we come down here and look at the mean for these different marine mammals, we see it's about 2.3 times higher than the allometric prediction for a eutherian mammal. The odd one out being the manatee, which is 30% of what we would predict. So it has an exceptionally low metabolic rate, which has a lot of physiological and ecological implications. So the increased resting metabolic rate, we believe it results from mitochondrial proton leak, which generates additional heat for thermal regulation. It is the basis for all endothermic heat production. The reason endothermic mammals have a high resting metabolic rate, 4 to 8 times higher than an ectotherm, is this increased mitochondrial proton leak, which is adaptive for offsetting heat loss. And in marine mammals, it's elevated even more, 2.3 times the allometric prediction for endothermic mammals. So an impressive adaptive increase in heat production. So when we talk about respiration, we refer to gas exchange between the animal and the environment, and this typically involves a gas exchange surface area, a thin epithelial layer that allows the diffusion of oxygen into the blood and carbon dioxide from the blood back into the gas exchange surface. Now we're going to be focusing on alveolar lungs today, but you could talk about a gill system as well. So the purpose of respiration is to maintain a supply of oxygen for aerobic metabolism and to eliminate carbon dioxide. Asphyxia is a term which is a life-threatening condition of reduced or interrupted oxygen absorption from the environment. And it can be result from a number of different factors. For example, a low oxygen environment, such as being at high altitude, breath hold diving in marine mammals, reduced gas exchange in the lungs due to disease processes, and that's what Terrie's going to talk about a little later today, in which the gas exchange surface area is reduced, thereby impeding the transfer, the diffusion of oxygen into the blood. Now, marine mammals are interesting in that they spend a lot of their time submerged in an environment which they cannot breathe. They cannot breathe water. They still have to come to the surface to breathe air. And so they end up dividing their time between 2 important resources: oxygen at the surface, food at depth. And they have developed a variety of behavioral strategies for maximizing the use of these 2 resources. And examples of that are depth and duration of diving, and the repetitive, a strategy for repetitive diving and rapid loading of oxygen at the surface. Pulmonary ventilation in animals evolved for gas exchange in air, not in water. And it evolved for a gas exchange in an environment which has a very low density and a very high oxygen concentration. So if we look at the density of water versus air, we see that water is 820 times denser than air, and it has only 4% of the oxygen of air. This is a highly dense, low oxygen medium. The high density of water makes it energetically impossible to breathe using an alveolar lung. It's just not possible. That's why marine mammals cannot breathe water. They never re-evolved the ability to breathe water. They maintain their alveolar lung from their terrestrial ancestors. And the low concentration of oxygen in water, even if they could breathe it, they couldn't absorb enough oxygen through the lungs to support the high level of aerobic metabolism, which is typical of an endotherm. So the terrestrial ancestors of marine mammals like the Archaecidea and the Protosirenidae, although they were terrestrial and entered the aquatic environment, they retained their alveolar lungs. They were never able to readapt the ability to breathe water. Here's a little schematic of pulmonary blood flow through an alveolar lung. I know you're all probably familiar with it. These are alveoli and alveolar sacs. And here we have deoxygenated pulmonary arterial blood coming from the right ventricle, passing through a capillary mesh that surrounds the alveoli, taking up oxygen, releasing carbon dioxide, and returning to the left side of the heart, where it is then distributed to the rest of the body. This is this is distinctive of an alveolar lung and differs from the avian lung. So the high metabolic rate of endotherms requires a very high level of oxygen absorption. And if we look at the alveolar lung, we can see some impressive statistics. The typical lung of a mammal is an elaborate array of airways, starting with the bronchi and ramifying into finer and finer airways, and a remarkable distance of fine airways that terminate in an enormous number of gas exchanging alveoli. The total surface area of the average human lung is something like 70 square meters, about half of a tennis court. It's a remarkable number when you think about it. And it's so large, it makes breathing under normal circumstances almost effortless. We don't even think about it. However, if that gas exchange surface area is reduced, oxygen uptake becomes difficult, and at about 10% of the normal alveolar surface area, gas exchange is no longer adequate. And if you're, for example, an emphysemic patient, you have to get supplemental oxygen at a higher concentration. The lung volume of mammals, unlike resting metabolic rate, scales linearly with body mass. So if we look at the body mass of a whale, it scales linearly with its size, just as it would with a smaller mammal. And that's shown here for a variety of marine mammals. The exception is the sea otter. It has lungs that are about 3 times larger than we would predict for a terrestrial mammal of that body mass. And there's probably an explanation for that. I'm not going to go into it today, but it is noteworthy. So these are some lovely lung casts that Jerry Koyman did many years ago. These are from a California sea lion, and I love these because they show the ramifying airways, which are white plastic, and then the veins, the pulmonary arteries, which are blue, and the veins, which are red. And it gives you a sense of the density of these airways and blood vessels. Marine mammals have a lung structure similar to terrestrial mammals, with a few exceptions, which I'm not going to go into today. But here are some electron micrograph of a small airway and alveoli. Here's a microscopic view showing the the airway and alveoli, kind of a spongy look. Again, a close-up of the alveoli, and here we can actually see capillaries surrounding the individual alveoli, and this is where gas exchange takes place. The interesting thing about marine mammals is that they are adapted for long interruptions of breath holding. It is, even though they breathe air, they are adapted to interrupt respiration for long periods of time. And yet they're able to maintain an oxygen-based metabolism and physiological homeostasis even while breath holding submerged. This is possible by a number of different adaptations, which you're all familiar with. The dive response, which is an apnea, a variable bradycardia, and peripheral vasoconstriction to maintain central arterial blood pressure. The earlier view of the dive response that originated with Scholander and Irving was based on a forced submergence, and it resulted in a very pronounced dive response. We now know that the dive response is much more dynamic. It is not an all or nothing response, and that it actually evolved to optimize the distribution and use of blood and muscle oxygen stores to maximize the aerobic dive limit. So rather than protecting the animal from asphyxia, normally it optimizes the use of blood and muscle oxygen. Its dynamic nature is shown in these two graphs, that publication that Terrie and I did, showing the relationship between the heart rate of freely diving Weddell seals in McMurdo Sound and bottlenose dolphins swimming voluntarily in a tank, I believe at Disney World in Florida. And here we have the flipper or fluke stroke frequency, which we take to be the instantaneous index of effort. So although we didn't measure exercise metabolism, the stroke frequency is directly related to exercise effort. So if the animal's stroking more frequently, it's exercising at a higher level. And what we see here is even when the animal is swimming submerged voluntarily, heart rate does increase as swimming effort increases. So what this actually is, now the resting heart rate of Weddell seals is up here at about 50 beats per minute. So this is a dive response. This is a bradycardia still, but it's a variable bradycardia, which changes with exercise effort. So it is an exercise response superimposed upon a dive response. Two ancient reflexes to protect the animal from asphyxia and to allow increased metabolism appear to be merged into this response, which is both a dive response and an exercise response. So bradycardia is less intense with increased effort, and that's necessary to increase convective oxygen transport to the muscles, which are now the active part of the animal, which is increasing metabolic rate. And it keeps them aerobic under most circumstances. So here is a little model showing a typical terrestrial vertebrate exercise response. Increased ventilation, shown here by these lungs, increased heart rate to transport oxygen in the blood to the muscles, or through the vessels to the muscles. This is shown for terrestrial animals having a fairly low hemoglobin concentration compared to marine mammals. And this convective transport of oxygen allows an increased metabolic rate, which is maintained aerobically. And we have a variable heart rate response, as indicated by this little meter. This was the classic dive response that was associated with forced submergence. This is artificial. This is the animal being asphyxiated in captivity, basically is what it is. Respiration stops, heart rate drops to a very low level. Blood flow is reduced to the peripheral tissues and organs. Now, the hemoglobin concentration is higher than a terrestrial animal. Oxygen transport is slowed to the muscles, which have an elevated myoglobin concentration. But the animal is able to adjust the convective oxygen transport. Well, here there's no option. This is a maximum dive response. When this oxymyoglobin runs out, the animal will go into anaerobic metabolism. In the free diving exercise modulated dive response, heart rate is variable depending on swimming effort, swim speed. And the amount of oxygen being transported to the blood, although reduced from the level at the surface, is still adjusted to maintain just enough oxygen from the blood to complement the oxygen that's in the muscle, which is substantial, to maintain aerobic metabolism. And as the muscle works harder, it needs to have extra convective oxygen transport in the blood, hence the increase in heart rate as animals increase swim speed while submerged. This was a study that was done in the 1970s. I was there when the study was being done. This was the study occurred in 1977 when I was down with Jerry and we were doing our Weddell seal ADL studies. This was Warren Zapol's group. And I think that this data is important before we get to Terrie's project, because what they did is they took Weddell seals into the laboratory and they basically forcibly submerged them. So this is not free diving. And they used radioactive microspheres injected into the left ventricle that then distributed throughout the animal's body to look at the percent change in blood flow between breathing at the surface and forced submergence. And you probably all know this, during forced submergence, most of the tissues and organs have almost a 90% reduction in blood flow. This is the peripheral vasoconstriction. So we have lungs, heart, kidneys, spleen, liver, pancreas, muscle. But there is no change in blood flow to the brain. It's maintained at the same level as it is at the surface breathing. There's no peripheral vasoconstriction. There's no change in blood flow to the brain. It's the only tissue that it maintains normal blood flow. It is not affected by the dive response. And that's very important to keep in mind. And because it's not reduced during forced submergence, we believe it is not reduced during free diving at all, where the dive response is even less intense. Now, what some of the implications of this I show you in this. This is a model that I did with my graduate students, Shane Kanatis, in 1999. We modeled the extraction coefficients of oxygen from the blood. The extraction coefficient is the percent oxygen extracted in one pass of the blood through, for example, a capillary. So it's the difference between, it's the arteriovenous difference is what it is. What percentage of the oxygen entering the tissue or entering the vessel emerges at the end, how much is taken out by the tissue. And we modeled this for, and remember, this is a model based on actual Weddell seal data, a variety of different data. We modeled it for different levels of exertion and got these plots. So we assumed that the saturation of arterial oxygen is 100% at the beginning of the dive. And by the end of the dive, it can drop down to pretty low level, 24 millimeters of mercury. What we found is that in most cases, muscle could no longer sustain aerobic metabolism when the saturation of arterial oxygen fell below 22 millimeters of mercury. So here we have the muscle bed. Here is the dive duration in minutes. Here is the percent extraction. So at the beginning, in these short dives, only 42% of the oxygen is extracted in the muscle. As we get into longer dives, we eventually get to 80%. And that's kind of the theoretical maximum. After that, you're going to drift into anaerobic metabolism. So here we see that we do hit the wall at the ADL, around 20 to 25 minutes. If we look at the brain, under all scenarios, under all levels of exertion, the maximum extraction coefficient of oxygen in the brain never exceeded 30%. Even down to 22 millimeters of mercury arterial PO2, there was still enough oxygen entering the brain relative to brain metabolism to maintain aerobic metabolism. The brain was never limiting to the ADL. Neither was the heart, and under most circumstances, neither were the splanchnic organs. It was generally always the muscle that ended the dive. That is when we exceeded the ADL. So this is very important to know, that the brain is not the weak link generally in aerobic dives. It does not set the ADL. We think it's generally skeletal muscle. Okay, I'm going to turn this back over to Terrie so she can carry on the discussion.
So that was great, Randy. Thank you very much. And so now we all are sort of on the same page as far as the basic physiology. And what I want to do with the rest of the time is just look at a few things that challenges this whole oxygen pathway and what happens when a disease process comes in to the scene. So this is where we start looking at how the heart and the blood and the brain are connected in our various divers. And I just want to point out a few key examples here. And if I had all of you in a classroom, like I normally am teaching this, I tell the kids, okay, take a big breath and imagine I've got a syringe and I'm going to take a serial or series of blood samples from you. And we'll look at how your blood is going to change in terms of the amount of oxygen that's contained in that, and just see how it changes as you're holding your breath. And that's what this graph is showing. It's actually showing elephant seals. Elephant seals are in the blue. This is by Jessica Meir. It's important to know that this is an arterial sample. The other samples that I'm going to talk about, except for the human, are going to be mixed venous. We have Weddell seals that are in black. You've got bottlenose dolphins that are in white here. And a human I'm going to show you in just a second. But if we were taking samples from you or any of these animals, what you're going to see is the longer the breath hold, the lower the PO2 is in the blood. Totally makes sense. If you get down to about 30 millimeters of mercury, this is when humans start to get in trouble, and we start to see, sort of this dysfunction in the brain in the sense of human shallow water blackout can occur. Something that's really interesting that's happened in just this last week has been a paper by Scott et al. This is a New Zealand group that decided to take an actual arterial measurement from a free diver. And so this is a paper, the link is down here. It's the Journal of Applied Physiology. and I think Jessica Kendallbar, who actually provided the paper to me. And it was a 2-minute free dive at 60 meters freshwater. And they had set up an arterial system with the radial artery, and they had 2 syringes just essentially taped onto this person, and had a diver on mixed oxygen, and at depth actually took a sample and then took a sample at the end of the dive. And after that 2-minute dive to 60 meters, you ended up with a post-dive sample of about 61 to 64 millimeters of mercury, excuse me, which is shown here by the star. And the interesting thing is that it took 2 minutes for that diver to get to that point. Wasn't near the human shallow water blackout, but there certainly was concern for it. And the marine mammals haven't even gotten started at this point. So what we find if we start looking at what the dolphins and the elephant seals and the Weddell seals are doing, these mixed venous levels can get very, very low for the animals. But when you're looking at arterial samples, this was arterial for the dolphins at this point. Here's the elephant seal here. You know, Randy and the modeling that he was doing was really not that far off in terms of sort of what these animals are willing to tolerate and what's going to cause a turnaround for the animals to start ascending from the dive. So there is a defense. What we don't know is how these animals, how the marine mammals are able to have a lower set point down in this range, 15 millimeters of mercury above, and humans are up here, you know, almost double that. Right now, and maybe some of you have more of a clue about this than we do, but it still is a mystery how these animals are operating and what adaptations are allowing them to get to such low levels. Well, one thing that we do know is that certainly marine mammals have lots of oxygen on board. So in addition to this dive response, the breath hold duration is enhanced by increasing the oxygen stores. We have blood stores, we have lung stores, we have muscle stores. And this is just giving you sort of an idea of the range of levels that we can see for animals. So here's lungs in black, here's the blood in sort of the dark gray, and then muscle oxygen stores shown in the lighter gray. But you get a sense that overall marine mammals have higher levels of oxygen on board as they are diving, or in preparation, sorry, as an adaptation for diving. But not for terrestrial animals. You know, we know that these levels stay low. It's not an adaptation that's necessary. We have oxygen pretty much available to us for use, for exercise, for rest, for thermoregulation. And the necessity for making these globins is just not there. And as a result, we maintain low levels. And for those of us that live at sea level and go to altitude, we know the difference between an adaptation and not in this case. Now, what we've done in my lab over the last few years is just try to look at some of the more vulnerable organs that are out there. Randy mentioned them at the end of his talk in terms of the brain, in terms of the heart, and trying to make sure that these critical organs are going to be supplied with oxygen despite the challenges that will be occurring, particularly as an animal is breath holding during a dive. And if you just do the comparative anatomical work, so in our lab, you know, just from road kills alone, we can get coyotes and mountain lions, bobcats and such. And then we've got marine mammals along the shoreline. And it's pretty easy to start to do these kinds of comparisons. And you can see just from the anatomy that something's very different for the marine mammals. If we look at some of the molecular proteins that are capable of storing oxygen, we see just neuroglobin levels. These are from a variety of terrestrial and marine. Here's mouse, here's bobcat, here's porpoise, pilot whale, and melon-headed whale. These are cerebral cortex samples in which we just looked at neuroglobin to get a sense of whether there was anything different between these animal groups. And certainly for the porpoise and for the melon-headed whale, we did see a signal, not so much for the pilot whale. And then as we got to the terrestrial animals, not at all. If we just look at, again, just the sheer color of the cerebral cortex, you see the difference between a terrestrial animal like a mountain lion down to a dolphin, which is shown in the lower picture. And what's become really apparent to us is the capillary density differences. There was work done by Karem and Elsner early on for harbor seal and comparing a harbor seal cerebral cortex to a dog. And they noted that there was a significant increase in the number of capillaries for the marine mammal. We're in the middle of doing these kinds of comparisons, have not finished it yet, but from narwhals to dolphins, we are seeing a general higher capillary density. And if nothing else, it corresponds to just the color of the brains when we look at them in a gross way. When we did an initial study of globins, and here all we were trying to do is look at different classes of mammals and say, are the brains different in terms of the proteins that would be capable of carrying oxygen, and compare them for terrestrial animals and for marine mammals. So here's a graph that we've published. This is resident neuroglobin or resonant neuroglobins. And so this can be cytoglobin, neuroglobin. It's a combination biochemical analysis. And we did it just as a percentage of the mouse concentration. We have yet to find a really good control to give us exact concentrations, the actual concentrations, even though we have it in terms of our analyses, the control values have escaped us so far. But in a general sense, in a relative sense, here's what the terrestrial animals are doing. You've got, except for the bobcat, which I cannot explain, but is a real value, most of the animals are down at mouse levels and fairly low. Hemoglobin concentration in the samples were low also. And you could divide out the deep diving animals, which tended to give us, low neural globins, but higher values when it came to how much hemoglobin was available. And then finally, the swimmers, where we tended to see these higher neural globins and not these extreme values in the hemoglobin values that we saw. It's tending towards a correlation with the capillary densities, as you might expect. And again, that's an analysis that's still ongoing. But at the very least, in a gross level, you've got a difference in terrestrial animals, swimming animals that tend to be moving fairly fast, and those that are the deeper divers. So this brought me to exercise physiology. So in the last year, we had to suddenly switch to online teaching, and I had to teach this exercise physiology course in the middle of COVID. it was just coming to fruition in the spring. And this was the question on everybody's mind. And so while we're running all these marine mammal globin studies, I've now got to teach exercise physiology and COVID biology, because that's what students are interested in. And that is what resulted in that paper that we put together, Randy and I put together, where we started to say, well, if divers are challenging the oxygen pathway, what is happening with COVID? And can we get a clue as to what's happening to a human patient based on what we know about marine mammals? And that's how that study actually came about. So we started with the normal oxygen pathway. And here again are our players, and said, well, really, what is happening here, both for a diver and the adaptations that allow those animals to tolerate, certain levels of hypoxia, and what appeared to be a problem for humans that weren't tolerating this at all when it came to this new disease. And the information is coming fast and furious. The great thing about COVID research is that most of it is accessible online for free. And it's because the medical community is just trying to get the information out for people. And so for the students and for the general public, and again, this is part of the reason that I put this study together, or put the paper together, is because it's sort of a public service announcement. And I've given this out to all kinds of people that just don't understand what's happening to their own oxygen pathways. And they've seen this, and you may have seen this on the internet, that just said, at the very least, we're starting with the lungs. And these are scans for a normal lung, a smoker's lung, and a COVID lung. In a very simple diagram, you have a sense that something is wrong with that initial compartment for the oxygen pathway. So this is what we know about this disease process. At the very least, people have actually heard the words, alveoli, and they've heard, trachea and pulmonary, and they're getting an idea that at least there is a problem with their lungs. And there's a problem with oxygen getting into the lungs, and this is creating a series of symptoms. So we have a good sense of it's starting at the lung, and we have a sense of the array of symptoms that are, manifesting themselves in these patients. And I just put together a pretty simple, combination of the things that are out there and things that are in the news, so that the students and even myself would start to understand what's really being hit here. And it was everything from COVID toes to all kinds of hemorrhages happening, the muscle fatigue that's occurring, cardiac arrhythmias, the acute heart failures, a lot of neural problems that were occurring, everything from just brain fog to confusion to a loss of taste and smell, headaches, dizziness, seizures, you name it, psychiatric impairments, and then some of the other organs, in particular the kidneys, being hit in one way by this disease. And the problem is that when you would see this in the news, it was like, oh my gosh, it's the heart getting hit. Oh my gosh, it's the kidneys getting hit. Oh gee, it's the neural problems with all of this headache and the rest of it, or fatigue. And in a sense, to me, it became easy to understand if you went right back to the initial problem, which is at one level, maybe a lot of this could be explained by there just simply isn't enough oxygen getting to these organs. It is likely not the only thing going on, because here are the mechanisms which I've put in the proverbial black box. But we've had, news on cytokine storms and microvascular inflammation, underlying preconditions creating a problem, these ACE2 receptors, and more and more. And we're going to learn more and more over the upcoming years. It's going to take a long time, I think, for people to really understand what happened. But at the very least, if you think of it in terms of you're just not getting in enough oxygen, despite all the things that are happening here, oxygen and the lack of it and a hypoxia problem can contribute to just about everything that is on this list. So it is my personal way of looking at this. I know that it's a lot more complicated, and I know that there's a lot going on, but it's very easy to explain to people that if this is your initial problem in your oxygen pathway, that these kinds of things can be predicted. And so in the paper, what I did was to give people a sense of what marine mammals have done in terms of the kinds of adaptations that these animals have to defend themselves against hypoxia, and indicate that with all of these issues that we're having, you suddenly realize what it takes to prevent that kind of damage or that kind of response for an animal that is trying to defend itself against low oxygen levels. And at the level of the vascular system, we already know increases in blood volume 2 to 3 times terrestrial, hemoglobin concentrations are up, O2 oxygen carrying capacity is up, we've got splenic contraction to increase hematocrit. The interesting thing is that physicians did understand that there was a problem in the transport of oxygen, and some of the earlier therapies that they tried were to increase hematocrit, not realizing perhaps that, at least for dolphins where it's been measured, that this Hageman factor, which allows for clotting, is actually missing in those animals. And as a result, you can have high hematocrits without the increase in clotting. And I don't think that that therapy is used that much anymore because of these clotting issues. But in terms of protection of the heart, increased myoglobin, increased buffering capacities, LDH enzymes, glycogen support, all there. Neuroglobins, capillary density increases, control of the vascularization, glycogen store for supporting brain tissue, perhaps decreases in temperature, and the kidneys too. So all of these organs that are some of the primary organs that are being hit by this disease have a lot of protection within marine mammals, and probably more than we even know at this point. And this is a graph that I used to show students just in terms of saying, well, how do we protect the heart or the brain? So these really vulnerable organs in the human body. What happens when things go wrong? And I'll explain why this was in exercise physiology in a minute. But the thing that struck me was that this graph was so pertinent these days, and I reproduced it in that paper because of how fast things go wrong when oxygen is not available in the mammal brain and in the mammal heart. You've got seconds where things are already happening at the level of the biochemistry of cardiac cells and neural cells. Within minutes, you're starting to get problems where you're entering into an onset of irreversible damage. And then with hours, you can have cell death and necrosis. This is fast. When you think of all the organs in the body, that within minutes, you've already got a scenario going where you are destroying cells and potentially never having them come back. This is a huge problem for these organs and being able to preserve them. What this inset graph is showing you is why this is an issue for these particular organs. So the tissues of your body, you've got determinate tissues, meaning there are tissues that when you are born, that's what you got. You know, at the moment, when it comes to cardiac cells, skeletal muscle fibers, renal tissue, and the brain, neurons, our ability to repair those tissues is fairly low. You know, essentially, you've got about 70 years before you can replace a myocardial cell. And that's the issue here. If these things die really, really quickly, your ability to replace them is actually fairly low. Intestinal villi, so there's a lot of GI problems too with all of this I haven't gone into. You can replace, and pulmonary alveoli to a certain extent, but by maturity, you know, maybe this is why younger patients are able to pull through a little bit more. They're still building these cells. By the time you hit maturity, we're pretty done. The indeterminate tissues, you know, are sort of shown here. We can make more red blood cells. We can make more endocrine cells and such. So you have a chance of repairing those things. But to me, this is frightening in terms of how fast the tissues go, and then the fact that you're not going to be able to completely repair them. And we see this, you know, stroke is just an example of what happens. And this is this is a personal journey at this point, but this is something that I saw in a family member that you don't want to see. And I just happen to know what happened because I was there taking them to the hospital. But this damage happened in a person that had all the signs of stroke, etc. Everything was happening really, really quickly in terms of locomotor damage, in terms of inability to talk, all the classic symptoms. You go to the doctor and I saw this and I knew exactly what the deal was because I knew this graph. I said, so is this going to get fixed? And they came back with, well, the brain has a wonderful way of restructuring. And it's like, yeah, okay, I got it. But it was interesting that the patients and stroke patients in general are disoriented and confused, don't know what is happening. And these are the kinds of things that you hear about in COVID patients all the time. And that's basically what this is showing. In COVID, it has the same scenario. The great thing, as I just said, is that remodeling can occur to some extent. You can have other parts of the brain take over and eventually you get most of the function back. And the same is true for cardiac tissue. Other parts of the heart will take over for tissues that have been damaged to a certain level. And here are the questions, and a lot of this is questions for me. Is the remodel as efficient as the original? Could be, but we don't know in the sense of COVID patients. And for athletes, are they going to come back compromised or are they going to come back stronger than ever? And that's where I really wonder about these longitudinal studies and the long haulers in terms of why COVID infection is scary for athletes, at least as I look at it. There are a lot of studies that I'm not going to go into right now, but it gives you a sense of really interesting science coming out because we're at that point where people are putting these 6-month studies together and dealing with databases that are based on millions of patients. And there was a couple that just came out in the last week or so. One was in Lancet Psychiatry, telling you that a third of the COVID patients, and this ranges from people that were relatively low level of infection to those that were in the ICU. But 33% of these people still had neurological and psychiatric problems 6 months later after infection. So this long haul is becoming important. There's a paper coming out in Nature. It's online as a preview right now, and you might take a look at this. But 6 months later, they're still seeing respiratory failure, insufficiency, and arrest, cardiovascular hypertension, cardiac dysrhythmia, vascular disorders, and again, the nervous system and neurocognitive disorders. Essentially, the entire respiratory pathway is still showing problems 6 months out. And then just as a last part for my exercise phys students, I used to show this graph that talked about sudden death in athletes that occurred generally in young people, but there are certain sports that are high risk sports for athletes. Basketball and football are the two big ones right now. There's a lot of discussion about fast and slow kinds of behaviors on the field. But basically, these are the two sort of difficult or challenging sports for the cardiovascular system. And this is where you see sudden death in young athletes or competitive athletes primarily. And here's a really final scary thing. We're already starting to see this in athletes. Young athletes, this was a Florida Gator basketball player, collapses on the field, promising young star. And they're bringing these athletes back. He had COVID. He has a myocardial issue right now, has not been able to play. There was a series of incidents with the National Football League. They started finding myocarditis in COVID survivors from them. These are two young football players at university and high school level, collapsed, died on the field. And the results are still pending in terms of the organ that was hit, but we'll see. And like I said, it's one of those things where, to me, there's a lot that's going on with the vascular system within the heart that I think is leading to a lot of what we're seeing in these long haulers as well as these athletes. So just to summarize, because I know we're running late, when pulmonary ventilation is reduced and dives exceed the aerobic capacity, marine mammals can enter into the beginnings of true asphyxia. And it resembles the reduced convective oxygen transport characteristics of what we're seeing in COVID-19 patients. The great thing is that nature's provided marine mammals with a whole arsenal of protections that will safeguard the tissue oxygen levels in the face of hypoxia. And I feel that it does provide a natural template for understanding some of the potential damage that can occur when we see oxygen deprivation in tissues in other mammalian species and during diseases that challenge this oxygen pathway. So with that, I'm going to stop sharing my screen and just say thank you for hanging in there the whole time.